Hello and welcome to the Ferguson Lab at Boise State University, where our goal is to understand how the genome works at the molecular level by single molecule live cell fluorescence microscopy. Our work is funded by the National Institutes of Health, and our lab consists of both undergraduate and graduate student researchers from physics, biology, chemistry, computer science, and engineering. So why physics? In the same way that early astronomers tracked the motion of the planets in the night sky, we now use fluorescence microscopy to visualize single molecules in living cells. 4 centuries ago, Tycho Brahe and Johann Kepler were able to carefully track planets and combine their data into a model of the universe including the laws of planetary motion, which then led to the universal law of gravity and the modern fields of physics and astronomy. In the same way, we now use fluorescence microscopy to track individual molecules inside of living cells to understand how the genome works in a new field of science called genome biology. In 2004, the sequencing of the human genome was just the first step. We now know all the letters in the Book of Life, but we don't yet know the grammar or syntax. In the same way that the Rosetta Stone allowed us to read Egyptian hieroglyphics by knowing ancient Sumerian and Greek, we must find some way of understanding the genome. In many respects, modern genome biology is like reverse engineering a 4 billion-year-old technology. The unlocking of the genome will lead to longer life, sustainable growth, and untold advances. If abused, it could lead to disease or even extinction. First, DNA is transcribed to RNA by the enzyme RNA polymerase in a process called transcription. When we sequenced the human genome, we discovered that only 2% of the sequence coded for protein. The other 98% was described as junk DNA. We now know that 25% of this non-coding DNA is a form of compression, turning our 20,000 genes into a blueprint for 200,000 and more. This is done through a process known as alternative splicing, where different protein coding regions are combined together differently or factorially to form an average of 10 different RNA sequences per gene on average. And then that results in the translation of different protein molecules by the ribosome. The process of alternative splicing is regulated by an enzyme called the spliceosome. The spliceosome is an ancient enzyme whose origins go back to early single-celled eukaryotes. Due to the complex assembly of both protein and RNA subunits, the process of splicing cannot easily be studied outside the cell, as we can for transcription and translation. So for this reason, single molecule live cell imaging technology is of critical importance in understanding the regulation of alternative splicing, which itself is a critical part in the understanding of the genome. So I'd like to thank the students who work in the lab. They are An Hune, Juliana Geltzer, Abigail Figueroa, Michael Poole, Garrett Tisdale, Iris Torres, and my collaborators in the biology department, Eric Hayden, and graduate student Jessica Roberts. I'd also like to thank my collaborators at the National Cancer Institute, Diana Stavreva, Dan Larson, Gordon Hager, and Tenika Lenstra, who's now at the Netherlands Cancer Institute. If you'd have any questions for me, please email me at mattferguson@boisestate.edu. Our work is supported by the Research Corporation for Science Advancement, Gordon and Betty Moore Foundation, and by a grant from the National Institute of General Medical Sciences. So again, I'd like to thank you for your time and attention, and I hope you enjoy the rest of your visit to the physics department at Boise State University.